Iron is a limiting resource for living organisms, and iron uptake with diet is usually very low. Sophisticated mechanisms evolved to recycle iron reserves. Iron availability is also a limiting factor for bacterial growth, thus it is not surprising that immune cells respond to infection adjusting their iron balance and iron cytoplasmic content to better support the inflammatory response. It is well known that macrophages can be functionally different in inflammatory condition or during the resolution phase of the inflammation. Those two macrophages are called M1 and M2 respectively. We know that M1 and M2 handle very differently iron. M1 tend to uptake iron, to sequester iron from bacteria, and also to better handle their need to respond to inflammation producing inflammatory cytokine. Vice versa, M2 tend to export iron to support tissue repair. We recently demonstrated that quercetin imprints an anti-inflammatory phenotype to dendritic cells, and we investigated genes involved in iron balance and demonstrated that quercetin-exposed dendritic cells express a gene pathway that supports iron extracellular export. Interestingly, quercetin is a potent iron phytochelant. Dendritic cells exposed to quercetin lose the ability to produce inflammatory cytokine in response to LPS, but this effect is gradually lost if in the culture media we increase the concentration of iron. We speculate that quercetin affects dendritic cells polarization, sequestrating extracellular iron and forcing dendritic cells to compensate iron balance using their cytoplasmic reserves. Once depleted from intracellular iron, dendritic cells express protein, including sleepy or HMOX, that block their inflammatory ability. With the present study, we propose for the first time a mechanism that links quercetin anti-inflammatory effect with immune cells iron balance, and we strongly believe that iron chelation is an interesting field to explore for treatment of chronic inflammatory syndromes.